Good morning, everyone, and welcome to Evercore ISI's Healthcare Conference. My name is Jingming Chen. I'm a member of the Biotech Equity Research team. We're super excited to have Andrew Hopkins today, co-founder and CEO of Exscientia. Exscientia is a pharma tech company at the forefront of the artificial intelligence space. So Andrew, welcome, and thank you for joining us today. So before I turn it over to Andrew for some remarks, I just wanted to remind our audience that this is an interactive chat. So please feel free to ask any questions through the chat box, and we'll try to get the answers. So Andrew, I'll turn it over to you. Exscientia is relatively new to the public market. So for those audience who might not be familiar with the story, can you give us a quick overview about who you are and what you do?
Absolutely. Great to be here today. We need to public markets, but not new to the field of drugs. And we've now shown that we have a company that's first proven how AI can be used to improve outcomes in oncology. The first sort of AI-driven clinical trial called EXAI, where we used computer vision to drive selection or which agents were used to which patients specifically through personalized medicine. That read out late last year, and it's the first time actually showed improved sort of statistical outcomes in how we can improve cancer therapy there. Also, first company to show that actually molecules can be designed by algorithm. The idea that we can move away from trying to integrate the wealth of data that we have externally into sort of medchem design, and actually now potentially design drugs much more efficiently, actually, using the help of AI-driven machine learning algorithms to actually generate the chemical structures and drive projects faster. What we've seen there actually is potentially projects generally running much faster, producing far less molecules than necessary to optimize to create a high-quality drug candidate. So I think Exscientia really has led the field, actually, in these AI first. So the first trial to show that AI can improve outcomes in oncology, and the first sort of new molecules now headed into clinical trials designed by AI, such as what we announced just a couple of days ago, which actually just yesterday, our Ignite AI trial, where our A2A antagonist now is being sort of precisioned, targeted particular populations there.
Excellent. So can you tell us a little bit about your team? What's the composition? How big is the team?
I think what makes Exscientia unique in biotech is actually the composition. We're nearly 500 people at the moment, of which about 45% each are technologists. Biotech. And so is for Exscientia precision medicines. We want to design the right molecule and select the right patients. That's what we use now AI approaches for. And importantly, then it's important to then think about how we do it. And that's where the technologists come in and helping us then define the new technology to help our drug hunters become more productive. The key to it is really trying to speak two languages. It's actually then sort of the language of drug hunting and asking the right questions, and then the language of technology where we can hopefully find new answers to help our drug hunters.
We often, when we talk about AI, we often hear how AI and machine learning can improve probability of success and allow you to design better drugs and faster and cheaper way. So how does your technology help you do that?
So it's helping us in two ways. Firstly, how do we select better patients? And actually, this is what we've been really excited about, what we announced in our earnings call a couple of weeks back, and actually the new clinical trial we announced getting CTA approval for just yesterday. What it's about actually is then using a combination of taking actual tissue patient samples directly from patients, and then actually understanding individualized drug response using a multimodal genomics approach, using high content screening, single-cell transcriptomics, single-cell genomics, and truly understanding at a molecular level what distinguishes drug response from one population from another at a personalized level. So this then is a technology that allows us then to really create our own unique gene signatures for drug response, and we can then use those in the clinic. So first, it's really about precision medicine is about identifying the exact patient population we're going to go for. And so that's a key part potentially of increasing the probability of success in the clinic. The other part of it potentially also designing precision drugs. Think of drugs actually as precision engineered technologies, where we want to actually understand all the properties we can target what was actually for the right target product profile, the right patient, and understand can we create the best medicine possible from the patient's point of view. This is where actually the use of sort of generative technologies, active learning, and techniques like this can actually create more targeted drugs. And we really excited about the molecules we're bringing forward, which we think actually shows incredible sort of PK, so we can really target that particular mechanism. And you'll see this actually in some of the new molecules will be announced as well in the next 3 months, how actually AI is helping us then design exquisite molecules.
Got it. So when you get a molecule, how do you validate that? How do you know if it's a good molecule, and how do you test that? Do you have a wet lab component as well?
Absolutely. About 40% of our company are actually experimental biologists, building everything from our patient tissue models, built from our biobanks, which we collected from over 70 different sort of clinical centers across Europe in our labs in Vienna, to also of large-scale biophysics, pharmacology screening that we have in our labs in Oxford in the UK. The reason being is that we want to control the quality of data we generate that we then bring into our projects and bring into our machine learning models. And it's really important for us actually to then understand and build these these unique translation models actually can help us then drive projects far better. It's a key part, I think, a key component actually of any sort of pharma tech or sort of tech-driven sort of biology company these days, is that one needs really to also to think about controlling and generating one's own data to feed into the algorithms.
Got it. So how long does it, just curious, how long does it typically take for you to, say, go from a target to a actual drug candidate?
So when we look at our sort of metrics, which we published, and every time we produce a new drug candidate, we publish a new metric. What we're looking at now is from sort of starting our sort of drug design project, when going after a selected project to go for, to potentially identifying the molecule that will become the drug candidate. We've seen metrics of around 12 to 15 months in our sort of from hit from first molecules to sort of identify industry standards, which usually in that particular part of the process around 3.5 to 4 years, for example.
Oh, wow.
What we're also seeing is the number of compounds we actually need to make and test potentially to go from was very first sort of initial ideas to the potential sort of drug candidate. And what we've seen then on average in the projects that we've got to date, which is about 7 drug candidates so far that are either progressing in IND studies or into phase 1, it's around 250 molecules, novel molecules being made and tested in the optimization procedure. And that's covering multiple potential drug series. To put this in perspective, big pharma would often make at least 2,500 molecules, sometimes 5,000 molecules in an optimization sort of procedure. And the reason why we think we're seeing these robust figures of our projects actually being very parsimonious, very efficient in exploring the space is the use of technologies like active learning. This is like a set of mathematics that allows us then to really try to ask a question, what should be the next molecule we should make and test that will provide us with the most information to drive a project forward, for the different properties that we need to optimize. And what's really interesting is that active learning often it really assumes that sometimes we start off projects actually with sometimes, not very predictive models. We sometimes start off with actually a paucity of data. It's actually a sparse data, small data problem often in drug discovery, rather than a big data problem. So the question we want to ask is how quickly can we improve our models and drive a project forward? And so that's a key thing about the mathematics we use in this space, actually. It's really thinking about drug discovery as a learning problem rather than a screening problem.
So do you take the learnings that you get from a particular drug and feed it back to your system to further train your algorithm?
And absolutely. And it's not just about trying to efficiently do that in a active process in sort of drug optimization. We actually think we're going to take our feedback all the way back into my patient select. So we really have a sort of this learning loop. We want to start with AI, start off with the cancer patients we want to treat, collect as much data on those individual patients as we can. We have over 70 different centers across Europe where we are building out biobanks from a wide range of different sort of cancers and patients, bringing along their sort of records of their treatment journeys and their health records, and then understanding at a deep single cell multi-omics level all we can about those tissue samples from those patients. And what's exciting about sort of collecting sort of these tissue samples and building models from them is that we believe it's some of the most relevant translation in the industry, not just homogeneous cell lines that we've grown from a single patient, and that all the different types of cell interactions. You have immune cells, cancer cells, stroma, you have clustering in 3 dimensions of these samples. This is why we had to build proprietary computer vision technologies to understand this. What it allows you to do then is to then understand sort of drug response on an individual cell-by-cell basis and really understand, is this drug, affecting a cancer cell or is it affecting an immune cell, or what's the difference between it? We are really excited by sort of this kind of technology. It's about bringing then sort of personalized medicine really into the heart of drug discovery.
I see. So just curious, how are what you're doing at Exscientia different from other AI drug discovery companies?
I think the heart of what we do is really the patient centricity and bringing that into an end-to-end process. We really do start with a patient at the beginning of it and drive our sort of portfolio and our approach towards what we believe are the most translational models in the industry. This is because we're driven by 2 factors. How can we make better drugs faster? So to make better drugs, we believe that we need to be creating better translational models. We have a failure rate of like 96% in the clinic in the industry. We believe that actually thinking about the human and the patient is the best model actually, but also faster. And this is where actually the concept of not thinking about AI or machine learning as one algorithm, but it's actually a whole suite of algorithms looking at different problems and really taking a sort of data first, AI first approach into trying to reinvent the actual process of drug creation. This is where fundamentally Exscientia comes from. It's not just about the projects we do, but to truly build a platform, it's about building the engine. It's about understanding and changing the process. And we fundamentally believe if we can change a process, that's how we can create a highly scalable company that can really make a difference to patients.
I see. And earlier you mentioned that there's a landmark EXAI study. And can you tell us a little bit about what are the key findings and how does that feed into your thesis?
Yes, EXAI is incredible, and I recommend people look at the paper. It was published in Cancer Discovery back earlier this year. So EXAI actually took the idea of taking sort of patient tissue samples and computer vision. And we did this at first in blood cancers. We're now looking to extend this into solid tumors, of course, as well, and there'll be more news about those further development shortly. And what we're seeing then is the idea then of taking that patient sample, and within about 5 days then being able to screen about 130 sort of different molecules, between 100 and 130 different sort of drugs, and see relative response of the cancer cells relative to all the other cells in that individualized tumor sample, and then actually be able to rank which actually potentially is the best drug for this individual patient at this time. But what was interesting, this was a prospective clinical trial. Everyone involved onto it had already sort of had no standard of care, and we've had multiple rounds of sort of previous therapy. We were failing all of the existing therapies. And what we were able to do then was to then prescribe what we think actually could be the right treatment option for that individual. Where actually the treatment option then was conducted purely by the physician. We saw that within 12 months, there was no progression. Everyone was sort of progressing. And what we saw then with the patients for which then they were selected, taken the drugs selected and recommended by the algorithms, is that we saw around sort of 25% of patients, even after about 4 years, were still progression-free survivors. And what's incredible about this is I think the ORR was 55%, the hazard ratio was 0.53. And this, I think, was the first time that sort of a algorithm-driven selection process based on machine learning using computer vision to really help and select and recommend it had really shown to make a difference in oncology by sort of the difference in progression-free survival between the treated and non-treated camp. This is an incredible approach, we believe. But what's exciting is that we see the potential of this going forward, actually, with patients, but also we're using that same technology now. We're expanding it into solid tumors. We are now using that same technology and bringing it all the way back into our processes. We use the same models for target discovery. We use the same models then for candidate discovery. And we use then the same models then for patient selection, as we announced in our earnings call, what we're doing with the EXScientia trial, which we announced yesterday. So what's really exciting about it is the process of bringing a patient into all stages.
Excellent. Yeah, I think that's really good news for patients. So let's now switch gear a little bit to the pipeline, since we just mentioned that. So first, congrats on the CTA approval for the 546. And can you tell us a little bit about this program, just a little bit overview what it is, and maybe speak a little bit to the trial design endpoint. So what questions are you trying to answer with this study?
That's a great question. Yeah, so this one was originally sort of generated using our sort of machine learning approaches, combining sort of biophysics and generative designs, etc. So it was rapidly generated as a molecule. But what we try to do with the endpoints, it's an open label study, 2 stages, multi-center, looking at safety, pharmacokinetics, pharmacodynamics, and efficacy, but all in combination with PD-1 inhibition as well. But the multiple endpoints, as well as safety and efficacy, is also looking at the biomarkers, looking at the new adenosine burden signature that we've identified, actually. And we will actually then be looking to make sure that we can also evaluate our biomarker signature in the trials, as well as evaluate the drug. And we believe that's important. And that really is actually part of a hallmark of thinking about Exscientia trials.
I see. So from a practical standpoint, so how easy is it to identify patients with the biomarker signature?
So we reduced this down, actually, using a multimodal now to a gene signature that actually we believe actually using counter-genomic technologies will be relatively easy to measure in the clinic. And actually was one of the key advances this year, moving from just sort of a high-content approaches now to truly multimodal, where we can reduce this down now to transcriptomics and genomics, which are much easier to deploy in the clinic.
Excellent. So I think to wrap up, Andrew, could you just give us a highlight of what should we be most excited about for Exscientia in 2023?
In 2023, what you should be excited about is us really demonstrating and bringing forward actually the concept of true precision medicine. In the new molecules we'll be bringing forward, and the demonstration of this actually will be expansion of our platform. We're really excited actually about the pipeline. Hopefully we'll be bringing forward to the clinic in the near future.
Excellent. Well, I want to thank everyone for listening, and thank you so much, Andrew, for joining us today.
Thank you very much.